Today's guests are James Taylor and Linda Walters. We join their conversation in the Faculty Lounge.
About 4 years now, has been looking at the health of the oyster reefs. And oyster reefs can be found all the way from Nova Scotia down to pretty much where we are in Central Florida, for at least the eastern oyster, Chrysostria virginica, the one that people like to eat on the half shell.
Yeah.
And over the past 50 or so years, the size of the reef and the health of the reef has declined dramatically. We've got maybe
Really?
1/10 of what we had in the 1940s.
Now, what do you attribute that to?
A number of factors. There is low levels of disease here in Central Florida. It's diseases that have wiped out populations in the Chesapeake and further north, but we just have them in low levels. They're protozoans and they can't handle the heat that well. So they'll die off in the summer. There's two main diseases you'll find, MSX and dermo. We don't have any MSX. Dermo we keep in low levels. We've been testing it with people from South Carolina, Department of Natural Resources. They've done the test for the last 3 years, and in the spring and in the fall. And we've had constant low levels of these protozoans. But
Is that a cyst or something that's in the...
Pretty much, yeah. So they do sections of it and they just count the number of these protozoans in it. And then they'll rank it from a really low number, well, from 0 up to the number 5, which means enough of these protozoans to kill it.
Where do they come from, the protozoans?
I honestly don't know. The disease, both of them didn't exist before like the 1960s.
Well, you know,
So-
sometimes protozoan cysts can come from runoff of cattle, from feces. There's cysts in infected cattle. So I don't know if that's out there or not.
I don't know in this case.
Sounds like a co-project there.
I'm just interested in finding out whether that's the cause of the decline. It seems like our numbers are between 1 and 2 for density, so not enough to kill them, but it's kind of just low level present. What I've been focusing on is the impact of recreational boating on the oyster reefs.
Oh, really?
So something a little bit different. If you go and travel down the lagoon, any of the passageways there, you'll see that some oyster reefs are very low, very brown in color, and they're totally covered up by water at high tide. That would be a pristine reef. That's what you expect. They're intertidal oysters. So at some point during the tide cycle, they should be underwater so they can filter feed, which is what oysters do. But what you'll see if you start looking is that there's some oyster reefs where you'll find huge piles of shells that are up to 1 meter above the tide line. And where is that coming from? That is coming from the boat wakes are pushing shells so that the shells are no longer part of the oyster reef. And so what they're actually creating are small little islands. That didn't used to be there. And we've done looking at aerial photos back from the 40s before there was a lot of high-powered boats out there, and you don't see any of these little islands that are very obvious in the aerial photos because they're very white. The shells get all bleached out when they don't.
You can't get on them or anything.
They're tiny. I mean, some of them are tiny, some of them are getting large, and we've seen them increase, and the number of them has increased, even in the 4 years I've been working on them.
Really?
And the biggest comparison people were worried about is, wind wakes could do the same thing because the water is very shallow. I mean, the average depth of Muskie Lagoon is 4 feet. Right. So, and it's a wind-driven system. So is the wind piling up the shells or the boats piling up the shells? And I didn't enjoy the hurricanes in this past fall that much, but we did reef profiles just by chance the week before the first hurricane.
I was lucky.
Just to get, the kind of, what is the shape of the reef.
Sure.
So the impacted ones would be this sort of thing. The pristine ones would be much flatter. So we did the before and after and didn't find very much difference at all.
Really?
So wind is for a short enough period of time, and it takes a good 30-mile-an-hour wind to start moving the shells around. where the single boat wake in our trials that we run can move, if you have shells in a quadrat, in a square, and we paint them so we know exactly which ones we're looking at, they can move 80 to 90% of those shells out of the area they started in, which is one boat pass.
Is that right? Does that hurt their health of the oyster?
Well, what happens is that oysters are gregarious, and their larval forms, they release gametes into the water, and they're fertilized, and they create small larvae that swim around for a couple weeks. When those larvae are ready to settle, they will preferentially attach to other oysters. So if they're the shell that they attach to, and that shell then gets moved so it's up above the tide line, they're going to die. And also just even the rolling over, we've made mimic little juvenile oysters, which are just little pieces of the clay, just to see what happens with all this rolling of the shells. Because the newly settled oysters are a paper-thin shell and can get damaged very easily. So we put little clay blobs out and found that the oysters, once they get tumbled because of the boat wakes, you'll see all these nicks and everything getting embedded into the clay, which is our proxy, and so mortality was very high just from that also.
That's got to have an adverse economic impact on communities like Grant and South Melbourne and all up and down the...
Not a huge number of people harvesting oysters, and it's much more recreational than commercial anymore. What the big concern is that these intertidal oyster reefs are nursery grounds for shrimp, for a lot of the commercially important fish like trout, drum, and all those sorts of things. So they are essential fish habitat. That's one of their main things.
Now, do you and your students take these oysters home and eat them sometimes?
Actually, I hate oysters.
I love oysters.
I wouldn't eat an oyster if you paid me. I'll go out and work on them in the worst weather, but you don't eat the oysters. Not
at all. Huh?
So,
do you do clams too? Now, clams got to be around.
There are clams around. They're subtidal, and there are more people who are harvesting clams than there are oysters, at least when they're available. We haven't worked on clams yet. I haven't had a student who's been interested in them.
That's right.
Where we're going right now with our oyster work, in addition to what I just talked about, was we've just found a new invasive species on our oyster reefs. It's a mussel called Mytella churuana, which is from South America. It was found up in Jacksonville in the early 80s, and they think it came in with ballast water from a ship, and it caused havoc in the intake pipes for a power plant up there.
Yeah, now that's a hard shellfish, right?
It's a hard shell, bivalve mollusk, So this mussel created havoc up there in the summer, and everybody was getting really nervous about it, and then it just died when it got cold. So it was they were very lucky. So what is it now? That was the early 80s. Now, 2004, one of my students found it for the first time in Mosquito Lagoon.
Oh, that's good and bad. It's good they made the discovery, but it's bad it's there.
Yes. So it took us over. We sent it to the U.S. Geological Survey because they've got an invasive species group that's actually in Gainesville, and the woman there took her almost 2 months to get it identified. Nobody knew what it was. She sent it out to all the experts. We just figured it was something that we just hadn't seen before. Not that it was an invasive, but I'm not a mussel expert, so. And
very few in Florida, I would bet.
That's probably true. So she finally got it identified for us, and I've got a graduate class now who's actually made a project out of looking for this. And in the winter months, in January, we were out there in some very miserable weather, and we're still finding them. Low numbers, but we're still finding them.
That can be a serious problem.
If it takes off, it can be a huge problem. And zebra mussels, you've probably heard of because you do freshwater work. And they have moved their way from the Great Lakes most of the way almost down to Florida already.
They really, those mussels can populate intakes and just shut down intakes. So it's a really severe economic impact.
Huge and ecological. They've displaced almost every native mussel that
we
used
don't
to be
want
there.
those.
No. There's another mussel called Perna viridis, which invaded Tampa Bay in 1999. It's a green mussel. It's really pretty. But it came, they'd found it in Trinidad in the early 90s, and somehow they figured ballast water introduction from somewhere between those two places.
Yeah, and I know you do some stuff with ballast water, too. I want you to talk about that to me a little bit.
The Perna has found its way from Tampa, where it did some more clogging intake pipes, and has made its way now within four years, all the way around the southern part of Florida, back up, and they found it off Jacksonville.
That's really a serious issue.
And large numbers of it in Jacksonville now.
It's really hard to kill those things. Yeah. Very difficult.
So once you've got them, they don't even think about eradication or anything.
Yeah, no.
Because they've also got larval forms that they can release millions of offspring that are going to populate many miles away. Mm-hmm. So it's more just control if they're a problem for you.
Now tell us how these species get here, and it seems I never understood that, how all of a sudden we're seeing a new species. And
ballast water is one of the big causes of introductions.
You better define ballast water.
Ballast water is going to be the water that you have keeping the tonnage of a ship constant. So big vessel.
I could do that. I could stand on one side or the other. Go ahead.
A little bit more than that. It's millions of gallons, and if they've got a lot of cargo like steel, they're not going to have a lot of ballast water. But when they're coming back the other way, they don't have the steel. They're going to fill it up with water from whatever port they were in.
And ports are nasty sometimes, real nasty.
But full of lots of funky critters.
Oh yeah.
That
like raw sewage.
Yes, things that like to live in raw sewage. Yeah. So the ballast water, what they estimate now is there's 40,000 gallons of ballast water released every minute in the U.S.
40,000 gallons every
per
minute?
Yep.
Yuck.
That's a lot, and it's just
They
a
don't
harbinger.
treat it or anything. They just dump it out.
Well, ballast water treatment is optional.
Oh. Okay.
So a ship owner can treat it. There are, just like in your drinking water, there's lots of different kinds of water treatments that they're trying to figure out for ballast water, using ozone, using chemicals, using electricity. Membrane, yeah, all sorts of things, but they all cost money.
Oh, yeah.
So if it's optional,
that's right.
may or may not happen.
Probably not.
Mostly not. The Coast Guard has been charged with the voluntary testing of it, and all they will do is go out and look at the salinity of your ballast tank, with the idea that if you're a good person, while you're out in the middle of the ocean, you will change out your water. So whatever you picked up in your port.
So you contaminate the ocean.
Well, it's not likely to get into anybody's backyard then. So you'll change out the water from the port that's full of creatures with open ocean seawater.
Yeah, I can understand.
And then when you go into the next port, the salinity will be higher. It'll be high, as high as seawater as opposed to whatever end. The chances of boats stopping to do that and actually just being in calm enough water that it's a safe thing to do is pretty limited.
Yeah, right. Are ports really low salinity all the time?
Some are, some aren't. I mean, it depends how much runoff there is. It depends just where it is. So it's not even always effective if the port salinity is the same as open ocean water, which is 36 parts per thousand.
Yeah, that's pretty big.
Yeah, so it's a pretty vague test, and they just use a little simple refractometer that I teach freshmen how to use. Drop of water on it and see what the salinity is. So there's a lot that needs to be done. I've actually been working with Debbie Reinhardt in your department.
She's acting chair of our department.
Looking at ferrate as a possible additive to ballast water to kill off whatever organisms are in there.
Yeah, no, that would probably work very well.
They've done chlorine in the past, but with all the residuals, it's an issue because the water is going to get dumped out somewhere.
That's
and
absolutely right.
The chlorine can just be the point source of pollution, killing every marine organism wherever it's dumped out. So they want something that's effective, but then whatever the word you use, it becomes ineffective.
Sure. The problem is only going to get worse because shipping is growing and ballast water dumping is probably increasing exponentially.
If the U.S. decides to label it as a pollutant, then I think the rules will change. But right now, with it just being voluntary, it's an issue.
Yeah. You know, I know you've done some stuff with coral reefs too. I want you
have.
to
That's
tell me a little bit. I've always been fascinated
third set
by
of
that.
research that I do. Again, it's human impacts on the marine environment. And what we've been looking at for about a decade now is what has the increase in seaweed, macroalgae, done to the corals and other organisms. What you used to find, and if you went on your honeymoon somewhere tropical, 20 or 30 years ago.
One of those I think I did.
You'd go to a coral reef and it would have been a coral reef. That would the main things you would have seen would have been sponges and corals and wonderful diversity. Now if you go to the Keys or pretty much anywhere in the Caribbean, once you go down diving or snorkeling, what you're going to see is algae. You're going to see a little bit of coral, but mostly you're going to see a lot of brown algae, in particular Dictyota. There are also some cyanobacteria.
Now those are nasty things.
They can be very nasty. A lot of the marine algae have wonderfully funky chemicals that are secondary metabolites to mostly curb predation on them. But what we've been studying
and kill consumers.
Well, usually the fish or whatever will spit them out.
Not humans.
So the humans aren't going to be eating the algae now. It's not terribly palatable. But what we spent last summer looking at was, can these chemicals in the algae, can they keep coral larvae from settling, or are they toxic to coral larvae? So once the algae's there, can nothing else come? Okay, so there have been people who are saying, well, it just wasn't a good time for corals, but they'll come back now. What we're trying to figure out is, do you need to clear off some space for those corals to come back?
Yeah, to make a...
So if you can just get rid of the algae, I mean, the coral larvae are there, but if they're killed in the process of trying to settle, then you haven't gotten anywhere.
Does coral grow any of the artificial reefs that are created?
It hasn't done very well in artificial reefs. Most of the artificial reefs you'll find, be it a submerged ship or a concrete structure or whatever, they have some, but they're usually more fish habitat.
Yeah.
So if you want to go, we're in a really good fish dive, an artificial reef is a good place to go.
Not for coral, though.
Not so much.
What do we got to do to promote coral?
The algae are blooming in part because they've lost some of their major predators, like urchins, and nobody knows why the urchins have died off. In some places they're starting to come back now in the Caribbean. They haven't come back in the Florida Keys. It was a waterborne pathogen. It's as much as they ever said about it in the 1980s. The other aspects, there's not much we can do there, but they are trying to reestablish urchin populations in some places. The Nature Conservancy in the Keys has been testing various ways of doing that. So we've been looking at whether urchins can't survive in areas with a lot of algae.
Yeah.
So even though they eat it, they may still have, it may be too toxic of an area for them to be when they're larvae or newly settled juveniles.
You ever relate any of that to pollution from like nitrogen and phosphorus?
That's the other side of it.
Yeah.
I mean, what causes any plant to increase in size or number is going to be those sorts of chemicals. So
really
just the increases in runoff and everything else, we've got huge blooms of these algae. So they're not invasive species for the most part, but they are there in a lot more abundance than they used to be.
Yeah.
So
Does all, the tourism, does that hurt when people dive to those reefs? Does that?
I think so. If I go to a place and spend a lot of money to go diving and all I see is algae, I'm not coming back.
No.
Even though I love algae.
But when the people dive, if they touch it or
it's not going to bother them.
Really, it doesn't?
No. Most of the chemicals in the algae or the cyanobacteria are actually held in vesicles inside of it. So you have to have contact, and you're not allowed to touch anything in the Keys.
Yeah, there have been some damage. I've read in the paper once or twice about ships running aground on.
Oh, yeah.
I bet you shudder when you hear that.
Well, you just know that it's going to be hundreds of years before things go back to where they were. But it's going to be a good place to study algae.
Yeah.
Because they're going to be our first weedy invasive
species. But really,
species.
hundreds of years?
Yeah. Corals are very slow growing. They'll grow maybe a centimeter a year. If you're thinking of a big, massive coral head, like a brain coral.
I'm thinking about coral hair. That's probably the coral hair I've got. Slow growing. No. So what's your prognostication for coral and whatnot? That's such a resource for Florida.
It is. It's a really important resource for Florida. Between all the human impacts and the fact of global warming and sea level rise, those are all negatives for our corals.
Really?
They say that within 50 years we may not have any coral reefs left that are in the kind of pristine state we'd like to see them in. So I hope enough groups start fighting for the corals.
Yeah, and
it's hard to get money to do research on coral.
Yeah,
which is kind of sad.
It is sad.
I've been very lucky and have had a reasonable amount of funding from NOAA and the USGS. I mean, none of the kind of big grants like you've had, but what they do, especially through NOAA, they have a National Undersea Research Center in Key Largo. And although they don't give you a lot of money to do extra stuff while you're there, they pay for everything.
Oh, that's
so they provide the housing, they provide the boats, they provide the dive training, the nitrox training, so you can stay down longer. And probably the coolest thing they have there is Aquarius, which is an underwater habitat. And they give out about
I think I've read about that.
3 awards a year to scientists anymore. The people at NASA use the habitat the rest of the time. 3 or 4 awards a year, and a group of 4 scientists will go down and live underwater at 60 feet for 10 days.
Have you?
Yes.
Tell me about that. Really, that's great.
It was very exciting. I mean, we collected the best data. I mean, 3 years later, I'm still analyzing the data. We got so much. Yeah. But it was the experience more than anything else. And it was myself, a professor from University of Tampa, and 2 of my graduate students. And I mean, there was lots of drama, as you can imagine. There were 2 technicians, so we aren't in charge of the safety at all.
Well,
I mean, other than our own personal safety, they were in charge of making sure the carbon dioxide scrubbers were working and all that kind of stuff, and the electronics were working. We were there just to get as many diving hours.
But you were underwater for
10 days. I actually gave 2 lectures at UCF from underwater.
You did?
I did.
What did you lecture on?
Well, I teach freshman biology in the fall, and they were my introductory lectures.
Oh boy, I bet nobody missed that class.
I have no idea. They could see me, but I couldn't see them. So it was a one-way camera. But it was very interesting. I mean, I got a lot of feedback on it.
Oh, I would imagine so. What kind of stuff? What kind of data did you collect down there? What did you study?
Well, a lot of data for doing research underwater, you've got to make it as simple as possible because...
No, I'm good at that.
Me too. Because you have so many things you have to worry about. We were studying at that point just the biology of some of these algae that had bloomed. And so we're just doing a lot of man hours looking at abundances and distributions. And one of the things I'd found out with the algae that bloomed down there is if little tiny fragments break off, something this sort of size,
small thing.
that's enough for a clone to be propagated. Within a few hours, it's put out rhizoids that can attach to at least 1 sand grain. That's enough to weight it down, and it just keeps growing from there.
Proliferation, big time.
So the little fragments are broken off by some of these very thin, weedy algae constantly. And so we were doing a lot of measures of how many fragments were in the water column, how fast were they produced, how much were they dispersing, so you could get some sort of idea of gene flow. And we were looking at photosynthesis rates also. So how fast were they growing?
How many square feet do you have to work in that thing? I mean,
it was basically the size of a big school bus. So not a lot of square feet.
Not a lot of square feet.
No. It was very tight quarters.
And there were 6 of you?
There were 6 of us.
That's like a submarine.
Pretty much. I think a submarine would be much bigger and easier. So there were 6 bunks, 3 on each side, kind of stacked on top of each other. Then there was the very small area with 2 computers and a table that 4 of the 6 people could sit at.
Now, how were the computers connected? Was it a hard wire running out, or was it a...
They have some umbilicals that had all the electronics that go from the habitat up to the surface, and there's a buoy at the surface that is a radar beacon and all that kind of stuff. So somehow it's all connected there.
Now, did you ever go out and scuba around in the reef and come back in? Or?
Oh, yeah. No, we were out all the time.
You were? You didn't spend the whole time in the...
Oh, no. That was driven me crazy.
Well, now when you were out, did you go up on top of the water?
No.
You were always underwater.
Our 60 feet was the highest we could go.
Ah.
Okay, so any diving we had to do had to be 60 feet or deeper.
Jeez.
So we were saturated with nitrogen, and that was like our 0 point there.
Yeah, because you get, I guess, sick nitrogen.
You get nitrogen narcosis. Yeah. So if you'd gone to the surface, you'd die. It's that simple.
Well, but you got to do it sometimes. So you had
at the end. We had our 10 days, and you'd spend at least 10 hours diving each day, which at the depth.
10 hours diving every day.
Every day for 9 days.
That you've got to be in pretty good physical shape to do that.
Oh yeah, you had to have a whopper of a physical before you were allowed to do this.
Well, I'm a whopper, but I couldn't do that.
So, but it gave you, instead of about 3 minutes at 100 feet, if you were diving from the surface, you had about 3 hours at 100 feet. And then you do your deepest dive first, which is standard for diving, and then you'd work your way back up to 60 feet and spend.
So you
the bulk
must be
of your
a
time
full-blown certified scuba diver then, huh?
I've done enough. There are a lot of people who've done a lot more, but I've done enough. But the last day of it is decompression. So anybody who works on an oil rig or whatever, you spend X number of hours getting the nitrogen out of your system as they're pumping air in, bringing it up to surface pressure. Yeah. And that, I will say, I loved every second of the first 9 days. That last day, the last 24 hours, I could have killed every single person who was there.
But you do that right in the
habitat. They've got it all pressurized, so they have locking kind of door that comes down, and they just change the pressure. And so slowly over the 24 hours, you're getting the nitrogen out of your system, and you could just feel
withdrawal. Huh?
Yes, and then all the things that didn't bother me so much while I was down there, because you get a little narked, you get a little silly, but not enough to be dangerous or anything. You could feel that going away, and then by the end of that 24 hours, it was, man, I just can't stand being in this little tiny space with these other 5 people.
You know, the claustrophobic issue, I have an issue with claustrophobia, but I was wondering about you down there.
There are some people that won't ever even consider doing it.
Yeah, I would.
I think, I had no problem with that. I have no interest in cave diving, where you get yourself diving into little areas that are no bigger than your body.
Yeah, if you want to continue to live, that's a good thing to avoid, I think.
Yeah.
No, I shouldn't say that. You lose so many good people that way.
There was enough space in this habitat that it wasn't a problem until
you plan to go back? Or?
We were awarded funding for the year, not this year, but next year.